going to end with a panel discussion of work group 3 that is going to try and bring some of these themes together that we've heard about regarding how are these many interesting mechanisms that we have heard about informing our ability to move potential treatments from bench to bedside, and what are the gaps and what are the obstacles for us to do that, and I'll turn over to both of you.
So earlier on Thursdays, you saw a back of the envelope description of what Workgroup 3 does, and you saw some lists of compounds which we came up with last year. And this was a particularly exciting time last night to have so many members of Workgroup 3 in the boardroom across the hall, and we got to check in with each other, have a very energetic discussion, and then really begin to discuss, now that we've come up with this list of compounds, a list of potential biomarkers or endpoints to look out for. What did it look like this year when the rubber hits the road and we actually had to go and talk to those doctors, try to find those studies, try to find the experience of individual patients and physicians who had been taking compounds, and figure out how is it that Workgroup 3 could best answer to its mission, which is to communicate these data to patients, to physicians, to companies, and do it in a way that, first of all, it does not give the impression that we know the answers, but really frame the questions so that they could be answered, and make sure that communication was clear and relevant. And so this spoke to the question of how do we take something that is a biomarker and translate it into an endpoint. This speaks to the title of this, or the initial title of this panel discussion, which is the challenges in trial design. How do we also get at the negative data when a study doesn't necessarily yield the information that we want it to, that's just as important as when we have positive information? And how do we make sure that those data are collected and communicated to the community as well, not only to prevent sort of the endless repetition of experiments that seem like a good idea and aren't useful, but also to let folks know what those outcomes are and have that inform the discussion further. And so I have a few notes here, but I can hand it over to you if you'd like to.
Make a general comment.
Go ahead.
Thank you, everybody, for staying till the very last minute, but it's important because I think what Kendrick are trying to do in Workgroup 3 in a general way is we want to help develop the landscape so that somebody walking by is like, oh wow, that looks like an appealing landscape where I can test a therapy or kind of learn something or advance the space. And the way that we do that is by trying to better understand what has promise for this area, better develop outcome measures, and also even better explore maybe some of the diversity within the patient population. So part of what I want to hear from the audience here is sort of as as we're mandated in work group 3, we don't run clinical trials, we don't discover or develop drugs, we just sort of help, like I said, arrange the landscape, what we can do to make that landscape appealing to drug developers or investors or other stakeholders to make it more appealing to you. So that's what I'm going to be asking you all, sort of what's going to be of interest.
And so what we did to take one of the things that I've written on the tops of several pages here is something that Florian started us off with, which is that patients are impatient and they want to know what they can do now. And I think one of the exciting things that we saw last night is that the physicians and the patient advocates and the scientists are also impatient. And we would like to get as much information to everyone as soon as possible. And so maybe this is and we've had, I think, this question sort of delivered to an audience several times. Troy did this yesterday as well, to ask for, what biomarkers and what endpoints would be most useful and most valuable to the community. Is that a good place to start off our discussion?
Yeah, I mean, I think maybe, so I guess I'm going to just sort of ask from the audience, like maybe you could call out some gaps to us. Let's start off with what's missing.
What would you like to see on Workgroup 3's agenda for next year? So give us some homework.
Or we're willing to take any questions, comments, we're very flexible.
Hi. Okay. I think one thing that would be interesting to look at might be TSPO. It's a translocator protein that's upregulating the brain after inflammation. It'd be very interesting to look at adult AMN patients and see if they have an increase in this. It's detectable via PET probes, which makes it very appealing.
For ALD patients, they were talking a lot about the blood barrier and about when getting things towards the blood barrier. Are they going to be working on that? And I know pretty much what happens is scarring or loss of gray or white matter. I'm not sure, I forgot. But loss of gray or white matter, are they going to try to have medicines to regain that and to have that back, so the synapses or the pathways of neurology, they work better?
So to your point, you're exactly right. One of the things that we actually heard about a lot is getting things across the blood-brain barrier, which has been an issue that this community has known for some time. But Workgroup 3 has never included that in our discussion.
When we, what part of what we do, we didn't have a chance to show in enough depth, is somebody brings us a compound and we do what we call due diligence on it. And we have specific questions that we ask of that compound to let it sort of go up or go down in terms of priority. And definitely one of the questions that we ask is whether it passes the blood-brain barrier. Yeah, so that has been identified as definitely a important attribute of a therapy that is part of our larger analysis of a particular agent. The reason why there might be, though, a compound that's very, very good at passing the blood-brain barrier, but it didn't make it, let's just say, all the way to the right side of the slide, might be because, well, it's a generic drug and we can't get anybody to invest in getting to develop it. It doesn't mean it will never be developed. It just makes it less appealing. So where we're trying to maybe deliver the value is put some of the biochemical features of a drug in the context of the commercial features of it. So we're trying to be realistic, but absolutely we do look at that attribute when we look at any.
Just to reiterate some of the topics that I think you have already highlighted and have been discussed, and I hope it hits the mark for Workgroup 3, that without mentioning unique biomarkers, there is, I think, for anyone interested in the drug development component, tremendous value in having good baseline data on an existing patient population where quality-controlled samples are available that have been taken under the right conditions, because a plasma sample or a blood sample or a cell-based sample not stored correctly may not be useful for the application that an individual wants. And the value of that, I think, is that dependent on the therapeutic entity one goes forward with, you can interpret it with regards to the perceived optimal patient population. And that means baseline, history, performance, how did the patient cycle, what was their clinical performance, how does that track with the perceived mechanism, and which samples can you use to best forecast which patients to include or enroll in the trial. Does that help?
Yeah, see, I think what this is making me realize in the discussion this morning, especially the previous speaker, is that we have a heterogeneous patient population. It's not just cerebral and AMN. And I think maybe we need to somehow think about slicing and dicing this heterogeneous population so that if there's a treatment effect, we won't miss it. And I don't know if that's really within our work group, but I know that very defined, narrow patient populations are going to be the best way to test therapy. And I'm not exactly sure how we can help move that forward, but I think that's going to be important.
So I have a question for somebody else who's going. For I don't know if this is necessarily the function of this work group, but is there any thought or focus on optimizing or improving animal models to screen? Just because obviously there's no clear animal model, it doesn't mean, I mean, of course there are animal models for several other disorders where just because it works in the animals, it doesn't necessarily work in patients. But it would be nice if there is a way to screen it rather than pulling surrogate markers from an EAE model or Alzheimer's or any other disorders, just for, earlier or quicker proof of concept and to be able to narrow down the therapeutics. What is the thought for that? I know this is like $1 million question.
No.
Probably.
Let me actually answer your question with another question. So I have some pharma people here. So let's pretend we're being introduced for the first time. I'm like, I want you to develop a drug for ALD. Sometimes your first question is, do you have an animal model? Are you still asking that, or now are you asking me, do I have a cell model? What question? Because it used to be the first question was, do you have an animal model? I feel like maybe people aren't asking that as much now. I'm looking in that corner. I just want to hear if it's shifting a little bit or if this is still so important.
I do not believe that has shifted. There are very good reasons to work in selected cell-based models, and in preparation for exploring a compound in the clinic, you often want to understand whether some of the predictions you make, whether they're PK/PD related or whether they're efficacy related, hold true in a hopefully predictive animal model. We all know that 50% of the drugs still fail because, well, our animals worked and all our cellular predictions were right. We just got it wrong and we could not align the data correctly. So I think you want both. It's not an either or. There's lots of advances, whether it's induced pluripotent cells, whether it's other cells, primaries, or whether it's animal-based models. I want all of them, and I would like to have all the data yesterday, so I'm most optimally prepared for when I go into the clinic. So if you could deliver that, that'd be fantastic.
Maybe Jim can also comment on that.
Yes, and I'd agree with that completely. I think that when the steps that Florian outlined this morning, the 3 steps in terms of mechanism, models, and clinical, I think, evaluation, certainly there are holes in all 3, but the last is, to my mind, still the biggest issue, is how are we going to effectively monitor the effect of a treatment in which patient population. I think that's still seems to me very difficult, depending on which type of therapy that we're going after.
So speaking from the patient advocate perspective, I feel a little bit mixed about the whole question, because when we first started this 15 years ago, we just kept thinking animal model, and then we realized in that time, now we've got a therapy in phase 3 that is not based on an animal model. I mean, you didn't correct any cerebral phenotype preclinically. So I guess what I'm saying is it's very clear, and obviously there's more examples of this, but here's an example in our indication where if we had said, let's not move to the clinic until we have a much better animal model, and I understand everything needs to be done in parallel, but you know what, when you're in a rare disease and there's limited resources, you can't do everything in parallel. So I guess I'm just giving a little bit of pushback, and I understand, like you said, you want everything and you want everything yesterday, and I understand that, but I think maybe as a community, we need to get a little bit more educated in a way to kind of push back intelligently there, saying, well, you can develop data preclinically in these systems that we have, and this is why they have value, and this is what we've proven so far, and then maybe marry that with some more homogeneous patient populations where we can get some early signals, not necessarily of efficacy, but just of activity. That's kind of, where I'm thinking.
So I'm 100% agree that the best experiments are done in patients, in the clinic. And if you can get there in a way that is risk mitigated, then can be a very fast route. In the absence of any preclinical data, it is often hard for smaller companies to attract the funding that will allow you to make that clinical progress. And if you don't get your PK/PD parameters in your phase 1, phase 1b trial aligned, you probably won't get the money to do anything else. So there's a clinical risk if you lack the data, and it's a gamble. So yes, patients first, clinical data and patients first. But the risk is if you fail there, that a good drug may not progress for the wrong reason.
So also maybe as part of our ongoing dialogue is to bring in the regulators a little bit more, because by no means do they insist that you demonstrate efficacy in a preclinical model, as you know. So I think we need to sort of understand that path, how to navigate it in our indication. Because of course you can develop PK/PD not in a phenotypic model, just physiologically. So I think sort of in my mind, we've talked about this even in terms of biomarkers. Maybe it's sort of like the next step is we do need to have a little bit next phase conversation with regulators to understand how they're thinking about this.
And to that point, as Keith is getting the microphone, one of the things that we did discuss yesterday was was bridging that gap between discussions of what research groups are planning in their experiments and what regulators would be looking for. And so if Workgroup 3 could essentially facilitate that conversation, that would be very helpful.
Yeah, I think one thing that that Workgroup 3 might do with Workgroup 2 is try and link some, to the extent that is possible, make a little bit clearer connection between the biomarkers of some kind and clinical outcome metrics, if there is any connection to be made. And I think, for those thinking about entering the clinical trial space in the different phenotypes or addressing specific symptoms, to try and be a little more concrete and user-friendly for the uninitiated to say, well, in AMN, some outcome measures that our group thinks are going to be helpful would be this, this, and this. These would be the biomarkers that might be relevant to AMN and may or may not be specific to these clinical outcome metrics. But I think if we could bridge that a little bit more concretely for people who don't know the space as well as we do, I think that might be a friendly way to invite people to look a little bit more closely at how they might do a trial. And because I think what many companies are forced to do is basically call an investigator or call, in many cases, several investigators to say, we got a drug, this is what it does, how would you guys run what are your outcome measures, what can we do? And I think that would be helpful to some of the companies. And maybe if someone wants to, anyone has thoughts on that, or if I think a few of you guys have probably been in a scenario where you've had to think hard about what kind of outcome metrics you might pursue. But so linking perhaps some of the work of Workgroup 2 and 3 might be helpful.
In my opinion, what we want and for what we want for preclinical models and what we want as endpoints completely depends on the drugs we are using. And for example, lovastatin and bezafibrate, we didn't need any mouse models. And actually, for lovastatin, the mouse studies were done after the patient studies were done. And then again, the patient studies were done to really get an answer, because the mouse data was inconclusive. And for bezafibrate, as I showed yesterday, the mouse model was totally worthless, because bezafibrate is not effective in mice. It's the wrong model. So for me, it completely depends on what we do. If we stumble upon a drug that has already been used in clinic, then you don't need a preclinical model. And for endpoints, there it also depends on what you're going for. If it's a drug that affects biochemistry, then it's clear what the endpoints are. It's biochemistry. If it's drugs, a lot of those drugs we're discussing in group 3 are the difficult ones, because none of these are associated with any biochemical endpoints. So those are the puzzles that we need to figure. So I think first we need so it depends on what we want, depends on what we're screening for and what we're testing.
So I completely agree about all models are wrong, some are useful, right? And so understanding the limitations and what you can use it for. I think on the endpoint side, I would love us to also be more open-minded about introducing maybe more outcomes of relevance to patients. And I'm really looking also here to patients, because I think we've been a little fixated upon certain things like walking speed and gait, and while, as we've heard in the last few days, there are many other topics that are highly relevant to patients. And the reason I think this is also important is because there might be outcome measures that are more tractable, more reversible than your good old spinal cord and gait and walking speed. And so neurologic, so bladder symptoms and urinary frequency issues, I think we heard so much about that yesterday. We heard about the endocrine and how relevant that is to patients. Just because there is currently some adrenal replacement possible doesn't mean that there's not still a window to modify, and there might be faster readout in some of these measures than in maybe balance and gait that might take 3 years. And I think we want to sort of, we want to ask FDA and the regulatory people, but they're really looking to us. So don't forget that really they're going to come back with questions to us and say, well, what do the patients say? So this is where we need more information from the portal. We need more input and contribution from patients online. And we've shown that patients can do this very quickly. We need to mobilize that whole population to go to FDA empowered to go after new targets. And I'm thinking of the TS field, for instance. So you might, with an mTOR inhibitor in that field, not have gotten your neurologic symptoms to change, but you changed the size of that tumor on the kidney, right? And that was meaningful. And it made you be able to move forward with a drug. So you have to, I think, be strategic and opportunistic. And we have a chance here to do that.
I agree with you. I just want to give an example that came up last night. We were given, we are always adding agents for review in work group 3. So one that came our way was an actually over-the-counter supplement called MitoQ. And we had kind of in-depth conversations with this company. And there's some good reports, anecdotal in MS, but then there's also some data in humans and some other neurological indications. But the bottom line was this. We ended up talking to the company, and they were like, you know what, this seems to really help fatigue in some of our neurological patients. So then the question is like, is fatigue a problem with individuals with AMN? Is it in men? Is it in women? Like, exactly what group of patients is fatigue an issue? And on top of that, the company was generous enough to say, we're willing to share with you 90 days worth of drug and 90 days worth of placebo. So here, just based on a conversation, we were sort of offered, I don't know, halfway towards a clinical trial. And the crazy thing was, I was not able to feed that into a clinical trial work plan where an investigator could say, 90 days, I know what signal I want to see at 90 days for a therapy that addresses fatigue in AMN, let's say. And the reason for that is because we don't know what the starting point is with fatigue in AMN. Then we don't know how we would necessarily measure it, and then what kind of a short-term signal we could see. But to me, this is like a potentially valuable resource that could give us a lot of information, even if it's negative information. And I feel obligated that work group 3 needs to be able, at the end of the day, have a pathway so that when we have a conversation like that and someone says to me, I can give you 90 days of drug and placebo, I'll be like, I know exactly who we need to talk to and what we want to measure. That's kind of where we need to go. And there's a lot of other symptoms besides fatigue, needless to say, that this relates to. And I think sometimes it's because the high-minded scientists are like, well, that's low-hanging fruit. But I think for daily, day-in, day-out patients, if you don't feel fatigue, that is very, very meaningful. So sometimes we need to remind ourselves that we don't really have to go for moonshots. Sometimes we just have to go for, New York City shots or what have you. So I just sort of want to it may not be as sexy, really, but I guess I want to remind, because everybody here is like, top of whatever it is that they're doing, but I think I just want to make and the companies as well, top, and just remember there's a lot of value in some of these symptomatic symptoms, and therapies do get approved every day in very difficult neurological diseases that address symptomatic issues, whether it's fatigue, psychosis, agitation, things like this, which are meaningful not just to patients but to their family. So that's what I want to say.
I have a question more for Group 4, patient engagement. I think it links also to Group 2 outcomes. A lot of our challenge is the statistical significance, no? And that depends on the N, the number of patients that you have. And just as a little snapshot, the fact that there is a person in Dr. Eichler's group that speaks Spanish has put almost 60 families in contact over the course of 8 months with more than 100 patients in follow-up. And we're translating the webpage and the portal to be to allow these people to come up. The world is huge. China, India. How can we accelerate this outreach? How can we accelerate both for education, but also for access for these people to report what's important to them and to help us develop the outcome measures and the biomarkers? They're eager to participate. I think most people here has this deep connection to this disease because of their own experience. This is not the only room where that happens. It's happening all across the world. How do we outreach to the world?
Would that be Keith? Maybe you probably thought about this.
Thought about the world.
You gotta learn a lot.
Probably thought about.
Yeah, I have one language. You're well. I mean, any input is for language barriers is welcome, and I think it's important. I mean, I think there was we had offers to connect us with the Japanese community already at this meeting, and to connect. Yes, and to. But I think it's an important next step. Yeah. We haven't, it has not come up recently as something we've specifically addressed. But there's, well, I don't understand. But it's something we should.
Can I ask a quick question? Oh, they all died, but I'm loud. As someone that, as you nicely put, deals more with the low-lying fruit, say, when I look at the work group list, I think there are tons of questions that we're kind of guessing at every day, like, how do I help a woman with pain, right? There are women that come and see us every 6 months for the last 2 years, and we're just guessing. None of those medications I really see on there, like, does gabapentin help with this or no? would an anti-anxiety medicine help with this or no? We just kind of blindly guess. And similarly with spasticity, I would say, you know, we have thoughts and ideas, but we're just guessing. And I think there's some on there for gait, but for pain, spasticity, and then because of my own bias, I would add all these boys that are on stimulants or other attention type medications, what's that effect? And for the men, you know, older people that are having some of those cognitive symptoms, should we be more aggressive? Would those same treatments that we already have be beneficial, but sometimes we don't do it because we're afraid or because I know, I mean, we use those in stroke patients and have benefit, but I don't think there's clear guidance right now as to how you treat some of those symptoms.
Well, I think, and to that point exactly, we were excited also in a conversation that we had with you in March to learn about Amitiza and some of these other compounds that really speak to quality of life issues. And so one of the things that we toyed with was coming up with essentially a new list of compounds that are basically the quality of life things, the things that just make getting through day-to-day life, gait, incontinence, these things. Can we go through this process with those compounds? And I think part of the issue is that whenever we would get together on our calls, we realized that we had a lot of curating to do on the list that we had already created. And so part of it is figuring out how to expand the list and then to keep up with everything that's there.
Because I think also if you do that, you might buy people in because it becomes so practical to be signing up to this because it's a real impact. You know, usually I at least think that when patients stop coming to clinic or stop engaging, it's because we're too slow. But these are things that we could make progress quickly because they're so easy to get, you know?
I really agree with you. I think it's all about back and forth. Like you give, you get. That's what ALD Connect is about. Hopefully we're giving, hopefully we're getting. I really agree with you, and I think maybe we need to talk about this like in a phone call, and that phone call will be dedicated. I don't know if I want to call it quality of life because it even goes maybe beyond that, or maybe quality of life is the ultimate endpoint beyond survival. I agree, and maybe we can coordinate with Work Group 4 and think about some ways to maybe test some existing medicines, maybe also kind of capture what's going on a little bit better. Because I agree with you, something like chronic pain or even mood disorders, which are not really described well enough, can be very impactful on a person, their family. But I think this is an area that we need to explore a little bit more systematically. I think everyone agrees.
Back to Patty's question. So I think that kind of social media is such a well-known space nowadays, right? So we heard yesterday, or out of nothing, right? So this YAC group, Young ALD Carriers, right? So do scientific research online through Facebook. So it's purely just resources. All teenagers, right? So all preteens, right? So know how to do this. So it's up to us to guide what questions to ask. But then there is so much knowledge how to do this, how to approach, how to engage. We can gamification, right? So now a new term, you can do it through games, through online. We did that video, remember, explaining ALD through cartoons, right? Unbelievable, right? So a huge response. So we can do all this. The question is resources and time. So if we can engage the same group of yaks, right? So people who are in the know. I discovered Twit recently because I didn't know that it's an individual thing. I thought I registered Neurobank Twitter tag, name, whatever, and anyone can do it. Apparently, it's just one-to-one. So again, but there's so much knowledge there, and there's so much interest and passion in this. So if we can just say, let's ask the right questions. Resources-wise, unfortunately, as you know, we're done with PCORI grant. So now we need to figure out how to support the portal. and everything else. I mean, luckily, my group has resources currently to do this, but in the long run, we need to do, I don't know, lemonade stand or whatever we can. But maybe based on the questions we're planning to ask in the future, we can engage sponsors to help us with this as well. Well, Stefan was, you know, swimming in canals of Amsterdam. Your queen was, yeah, for LS research. So.
So I think I sort of heard messaging that we need to continue to kind of refine our approach towards endpoints and biomarkers. We need to coordinate a little bit better with work group 2 in terms of some of those outcome measures. We probably need to coordinate better with work group 4 in terms of this virtual collection of who's experiencing what where, and then querying outcome, and querying that population, but almost as a separate, almost as a separate thing. And then at the same time, we need to sort of remind ourselves that there's a lot of symptomatic, maybe not so sexy symptoms that we need to continue to understand them better and test therapies better so that we can at least have some conclusions that can be shared within the more broader healthcare delivery community and the patient community. And there was a handout number 1, which Raz made a bunch of copies for, and do you still have them?
Yeah,
okay.
They're out on the registration table.
Okay, on the registration table, there's a handout called handout number 1. And what it is, it's a little fill in the blank with maybe about 7 or 8 boxes to fill in. So when you all leave here and go about going to lectures, reading articles, thinking about your own research or your own circumstances, and you encounter something that might be of interest, this handout number 1 is a way for you to register that agent. And it just requests a series of information attributes about that agent. You're not expected to fill it all in. And then you would turn it back into us, and we would add that to our list. So I just really want to advocate that we're all ambassadors for this disease, and part of that is that we're all detectives looking for clues, and handout number 1 is a way to bring the clues back to home base.
I just wanted to tell you how exciting it is to present the slides from Workgroup 3. And the evidence for this is that I put up the goals, and then I put up a list of compounds that we're reviewing, and I see all of the cell phones come out from the audience. And regardless of where we are, if it's in Omaha or in Baltimore, people are taking pictures of this list because they feel like it is something that's actionable, it's something that they can research themselves. And so, getting feedback from the entire community about how to, feedback from the community about how to expand that list, what else to add, what's important to you, that actually is something that we would be excited to add. So, it would be fantastic to have a lot of Workgroup 3 ambassadors to help inform us. And please fill out that sheet. If you feel like you're not comfortable filling out that sheet, send Rachel or myself an email and just say, hey, I heard about this, I read about this. I'd love to have some feedback from the expertise and the professional input that Workgroup 3 has access to. In addition, we're excited to in the next couple of weeks, we're going to be putting together an abstract to present at the AAN meeting in Vancouver this year, or next year, about the Workgroup 3 experience of evaluating compounds with this algorithm. Because what we realized at the ULF meeting is that people were coming up to us and saying, well, hey, I'm not part of the ALD community, but I'm interested in using that method, that homework sheet, that system of discussing these compounds for my own indication. And so we got a lot of feedback that people wanted access even just to the method. So we're going to be putting together that abstract and presenting that to a larger community as well. So we welcome your participation.
Yeah, so on behalf of Kendrick and myself, thank you both. I mean, thank you all very much. He's wonderful. So thank you.
Great. Thanks so much, Kendrick and Rachel. I think